Welcome to this series that will introduce you to diversity interactions modelling. Episode 2. What is diversity interactions modelling? Diversity interactions models jointly model the effects of richness, composition, and species proportions on ecosystem function. So how do they do that? First of all, the type of data that is needed to fit a diversity interactions model is the proportion of each species in each plot or experimental unit and the ecosystem function response. The data will look something like this. The first row of this data shows a monoculture of species 1. Second row shows a 50-50 binary mixture of species 1 and 5, while the third row shows an equiproportional mixture of all 5 species. So each PI is equal to 0.2. Diversity interactions models are multiple regression models. The response variable is an ecosystem function. The predictor variables are the species proportions and their interactions. And there's no intercept because the species proportions sum to 1, and the model would be over-parameterized if there were an intercept. This is the structure that a diversity interactions model takes, where ID stands for identity effects. Y is the ecosystem function. ID and interactions include the initial species proportions and their interactions as predictors. Block or treatment predictors can be included in structures, while epsilon is usually a normally distributed error. Here's an example of a diversity interactions model. The beta i's are the identity effects. The delta ij's are the interaction parameters. Theta is a nonlinear parameter. And alpha k is a block effect, but other treatments or covariates could also be included here. In this equation, there is a unique interaction parameter for each pair of species, but more parsimonious versions of the interactions are possible. For example, species interactions might be dictated by functional group membership. In the data shown here, there are 2 functional groups, one for the 3 blue-coloured species and one for the 2 orange-coloured species. Let's take a look at fitting a functional group model for the data shown here. Here's an example of a diversity interactions model where interactions are dictated by functional group membership. There's an identity effect for each species, which is the name we use for the beta coefficient of each species proportion. With 5 species, there are 5 choose 2 equal to 10 possible pairwise interactions. Here we group them according to functional group membership. Remember that species 1, 2, and 3 make up functional group 1, while species 4 and 5 make up functional group 2. So instead of 10 pairwise interaction terms, which you could have, here we have 3 interaction terms. Delta FG1 is for the interaction for any pair of species in functional group 1, so any pair of species 1, 2, and 3. Mathematically, we sum the 3 PI/PJs that involve species 1, 2, and 3. Delta FG2 is for the interaction between the 2 species in functional group 2, so the interaction between species 4 and 5. The summation term describes this interaction term generally, but because there are only 2 species in functional group 2, it simplifies here to just P4 multiplied by P5. And delta FG12 is for the interaction for any species from functional group 1 with any species from functional group 2. So any of species 1, 2, and 3 with either of species 4 and 5. Here, we are adding up the 6 individual PI/PJs that involve 1 species from each functional group. This functional group model is more parsimonious than having an individual interaction term for each pair of species, because we have only 3 instead of 10 interaction parameters. We could also have, for example, that all interactions are the same. You can compare the various forms of the interactions using standard model comparison techniques to decide what form of the interactions works best for a given data set. Here's the functional group model equation again. Here are the identity effect estimates. There's quite a bit of variation across the species identity effect estimates. The first 3 are identity estimates for the functional group 1 species and are higher than the last 2, which are identity estimates for the functional group 2 species. Here are the interaction estimates. The interaction between pairs of species from functional group 1 is not significant. There's a strong interaction between the 2 species in functional group 2. And there's a strong interaction for pairs of species with one from each functional group. Here we can see the raw data again. Can we see patterns identified by the fitted diversity interactions model in the raw data? Well, species 1, 2, and 3 have the highest identity effect estimates, 37.0, 30.3, and 24.3. While species 4 and 5 are the lowest, 17.4 and 13.6. And we can see this in the raw data monoculture points. The identity effects also contribute to what happens in mixture, not just monocultures. So it's the combination of both the identity and interaction effects that determine outcomes in mixture. In the raw data, we can see that pairs of functional group 1 species do not have strong mixing effects, and this is reflected in the delta FG1 estimate of 5.3. In the raw data, we can see a strong species 4-5 interaction in the data, reflected in the delta FG2 estimate of 40.4. And in the raw data, we can see a pattern that mixing species with one from functional group 1 and the other from functional group 2 is beneficial. And this is reflected in the delta FG12 estimate of 53.5. However, there's more to understanding the output of a diversity interactions model than just examining the coefficients. We can predict from the model to understand the joint effects of richness, composition, and species proportions on ecosystem function. Thanks for watching episode 2. You can tune in to episode 3 to find out more about how to predict from and interpret a diversity interactions model.